Photosynthesis functions as a highly mechanical manufacturing process, one that physically builds mass out of light and thin air. This diagram shows how that manufacturing process is split into two distinct operations. On the left, the light-dependent reactions harvest solar energy. On the right, the light-independent reactions, or Blackman's reactions, actually build the sugars. The purpose of those light reactions is to act as a power plant. They generate what we call assimilatory power, specifically ATP and NADPH, which is shipped across the divide to fuel the dark reactions. The universal engine driving those dark reactions is the Calvin-Benson, or C3 cycle. At the center of that cycle sits a single crucial piece of molecular machinery. This schematic illustrates the enzyme RuBisCO, represented here as a gear. It is the most abundant protein on Earth, and it performs the heavy lifting of carbon assimilation. Its job is highly specific. RuBisCO takes a 5-carbon molecule called RuBP and merges it with a single 1-carbon molecule of CO2 pulled from the atmosphere. That merger creates a highly unstable 6-carbon intermediate, which instantly splits to form 2 stable 3-carbon molecules known as 3-PGA. Mastering plant metabolism requires tracking the exact mathematical cost of running this RuBisCO engine. More importantly, it requires understanding of biochemical flaw in the enzyme structure, an inefficiency that forces plants to adapt their entire cellular layout to remain productive. This cycle map breaks the C3 engine down into 3 distinct phases: carboxylation, reduction, and regeneration. If we drill down into phase 2, reduction, we can see exactly where the plant invests its assimilatory power to turn those newly formed 3-PGA molecules into actual sugar precursors. The chemical transaction here is strict. To achieve this reduction, the plant must spend exactly 2 ATP and 2 NADPH for every single CO2 molecule it fixes. The process continues into phase 3, regeneration. This is a complex carbon chain transformation where the plant must constantly rebuild its initial carbon acceptor, RuBP, or the entire cycle halts. Finalizing that regeneration requires one last energetic payment, a drop-in of 1 additional ATP per fixed carbon to prime the newly rebuilt RuBP. This energetic ledger lets us tally the final mathematical accounting of these transactions. Looking at the microcost on the left, fixing just 1 CO2 molecule costs a total of 3 ATP, 2 in reduction, 1 in regeneration, along with 2 NADPH. To see the macro goal, we extrapolate that math. Creating a single 6-carbon hexose sugar, like glucose, requires 6 full turns of this cycle. That sets the baseline for C3 plants: 18 total ATP and 12 total NADPH to synthesize 1 glucose molecule.